Hello everyone. I'm Dr. S. Rajesh, Associate Professor in Biotechnology from Center for Plant Molecular Biology and Biotechnology. Today we'll be discussing on construction of recombinant DNA molecules and bacterial transformation. So this is part of lectures on genetic engineering and its applications. Recombinant DNA technology is basically a technique where the selected DNA of a donor organism is introduced to combine with the DNA of any of the recipient organisms. As the result of this recombination, the uh, recipient host or the genome acquires the genetic abilities of the donor through the recombination process. So the recombinant DNA technology is also popularly called as genetic engineering. So the DNA sequence which is used in the construction of recombinant DNA molecule can basically originate from any species. Practically it can be from any species or any organism. So using this recombinant DNA technology, the DNA sequence can be either created and introduced into any of the living organisms. Now let's see the recombinant DNA in proper. Recombinant DNA is a DNA molecule which is composed of sequences derived from different sources, and these composite DNA molecules comprises covalently linked segments from two or more sources, which we refer as recombinant DNAs. So this technology is also referred as DNA cloning or genetic engineering, used to apply for generating large number of identical molecules or clones in a host expression systems. So the DNA can be manipulated in an organism, which would program, say for example, the bacterial cells, it would program the bacterial cells to make the important proteins or the recombinant proteins, which may include expression of antibiotic resistant genes or the biopharmaceuticals or production of some therapeutic proteins or industrial enzymes in the bacterial host cells as an expression system. Inserting the desired gene into the genome of host is not as easy as it sounds. Although gene cloning is very popular term nowadays, it's routinely used in biotechnology, but the experimental procedures involving cloning of a foreign DNA into a vector system, mobilizing them into a host cell system, although it sounds easy, but practically it's not so. It involves the selection of a desired gene for administration into the host cell system, followed by selection of a perfect vector system. It can be a bacterial based vector like plasmids or a viral based vectors like phages that can be used to clone the DNA fragment or integrate them into a vector system and mobilize the recombinant DNA into a host cell system for production of recombinant proteins. Now let's see the construction of recombinant DNA in detail. So basically the cloning of DNA in any organism involves 5 different steps or general procedures involved for cloning and constructing a recombinant DNA molecules and subsequently transforming them into a bacterial host expression system. The first and foremost step is isolating DNA and cutting the DNA into fragments. So this is done by using a specific enzyme called as restriction endonucleases, which would cut the DNA at precise locations by recognizing the appropriate recognition sequences. And this deploys the enzyme called as restriction endonucleases, which we also refer it as molecular scissors. The second step is selecting an appropriate vector system which is capable of independent or autonomous replication, which can also multiply along with its own genome. It can also multiply the DNA of the cloned DNA from any other sources. So these DNAs are pieces of DNA which are used to carry the foreign DNA are referred to as vector system or cloning vectors, and they typically mean here we mean the plasmid DNAs are viral based DNA vectors. The third step in DNA cloning is joining together the two fragments. One is the vector DNA fragment, the other one is the insert DNA fragment, which would code for genes of any of the agronomic traits by using a specific enzyme called as ligase, the DNA ligase. So commercially, we have two sources of DNA ligase, E. coli and T4 DNA ligase, which we'll be seeing subsequently. So here you can see a cloning vector, plasmid is basically is going to be used as a carrier molecule to clone the foreign DNA. This can be done by restricting the vector as well as the foreign DNA with the restriction endonuclease that would open up the plasmid and make an fragment that is amenable for cloning in the vector system. So finally, once you ligate with the ligase enzyme, you would get a chimeric DNA or a recombinant DNA. The fourth step is transformation and recovery. Here, the constructed recombinant DNA is going to be mobilized from the test tube or under in vitro condition to a host cell for biological replication that will provide enzymatic machinery needed for copying of the cloned DNA along with the plasmid DNA or the viral vector DNA. So this would allow the cells to multiply the clones in a rich medium. Most possibly, we will be using the LB medium or SOC or 280 medium for bacterial multiplication. The fifth step or the last step in the construction of recombinant DNA technology and transformation is selection of the host cells which would be carrying the recombinant DNA. So we have steps to screen those cells which contain the recombinant and non-recombinant or the transformant and non-transformant based on the visual screening or enzymatic digestion of the cloned DNA fragments. So we will see in detail about the 5 different general procedure in this lecture. The first key player for this cloning procedure is the vector, which is the base for formation of a recombinant DNA. And the vector can be a plasmid, an artificial chromosome, or bacteriophages, which are widely used in molecular cloning procedures. It contains small segment of DNA that would have a signal for replication. You can recollect I was referring that this vector system should be capable of autonomous or independent or self-replication without depending on the chromosomal entities. The basic characters of these vectors are ideally they need to be capable of replicating in a host cells and they must be very small enough and for manipulation and easy to isolate using the laboratory procedures, and they should have some selectable marker which will help in identifying the presence of a resistant gene and differentiate a recombinant from a non-recombinant or a transformant from a non-transformant once it's mobilized into a host cell. And these vector systems should have convenience of an array of restriction enzyme recognition sequences for inserting the DNA fragment. So that's we have seen earlier as multiple cloning sites or polylinker sites, which have a choice of restriction unique cleavage sites. So now let's see in detail about the plasmids. So these plasmids, basically, they are capable of passing between bacteria, either for a specific role played for during fertility, like we have F plasmids that would move from F minus to F plus to F minus strain. So F minus would be a recipient of the fertility factor. And we have some plasmids which have genes that would impart resistance to some of the xenobiotic compounds like antibiotics or hydrocarbons. So these genes are present in the plasmid bacteria. Plasmids of the bacteria are beneficial genes which would give the bacterial cells some kind of advantage with respect to resistance to antibiotics or virulence factor to challenge upon the host cells. Say, for example, agrobacterium, it carries a Ta plasmids and a virulence region in that makes it pathogenic to attack the dicot plants where it can produce opines and nourish and multiply its cells through the infection. And some of the bacteria possess a beneficial genes for nitrogen fixation in symbiotic microorganisms. So these plasmids can be engineered to contain beneficial genes. So, what are those essential genes which we target is primarily there are three regions which we are needed, which we need to use it as a vector system for cloning purpose. The first one is origin of replication. It's a piece of a DNA sequence which would show the bacteria where to start the reaction of replication or copying of a plasmid from. So that region is referred as ORI, or origin of replication. The second region is called as multiple cloning site, which will have an array of host recognition enzyme recognition sequences that can be used to cut the plasmid DNA and open up to clone the insert DNA here. So this MCS region is also called as polylinker region. The third region is the selectable marker system. Appropriately, this would be an antibiotic resistant gene, which helps in identification of the recombinant molecules during the screening process. Now let's see an example of a kanamycin resistant gene, which is part of an E. coli TN903 transposon. Kanamycin is an antibiotic, which is a neomycin based antibiotic, which binds to the ribosome and normally prevents from translation of the proteins. So, contrarily, the kanamycin resistant gene, which is also referred to as NPT2, which is a neomycin phosphotransferase coding gene, that enzyme would degrade the kanamycin antibiotic, making it inactive and allow the host cell to survive in the presence of resistant genes in the plasmid. So that can be a good system for screening of the recombinant molecules. Having seen the key players in the recombinant DNA technology, the first one being the plasmid as a vector system. We will also get to see that other two enzymes. One is restriction endonuclease for cutting. The next one is ligase as a glue for pasting the DNA into the vector system. The first one step in the cloning procedure is DNA isolation. So DNA isolation can be performed by adopting any of the genomic DNA isolation procedure. In case of plant, we can isolate the plant genomic DNA by a very popular method called as CTAB method from Doyle and Doyle. And in case of bacterial DNA, we have procedures to break open the cell wall of bacteria by using lysozymes, or we can make detergents to break the cell wall and release the genomic DNA. And subsequently, purification can be attempted using a differential centrifugation by applying the denaturants like phenol, chloroform, isoamyl alcohol. Based on the densities, they help in separating the pure form of DNA and precipitate them and finally use them as a pure DNA for cloning purposes. Once the appropriate DNA or a target DNA is isolated from the host cells, that can be used to clone in a vector system to construct a recombinant DNA molecule. So how do we proceed further? We will be using an enzyme called as restriction enzyme, which is an endonuclease, which recognizes the specific sequences in the DNA and cut at those specific points. So we have varieties of restriction endonucleases commercially available. We have more than 3,000 restriction endonucleases available on the market with differing host recognition sequences. Most of them cut at a specific palindromic sites in the DNA, and they generate fragments which may be a blunt-ended fragment, or it may be a sticky or overhanging fragments. So here you can see the restriction enzyme cuts and produces the blunt-ended fragment in this case. That's from an enzyme from a source called as Haemophilus aegypticus 3, HA3, whereas the Providencia stewarti and EcoR1 enzymes, they produce protruding ends or sticky ends. And comparing the protruding ends with the blunt ends, protruding ends or sticky ends are very much amenable for cloning in a vector system. The second procedure or second step in the cloning process is joining the DNA fragments using the ligase, DNA ligase. So this is yet another fundamental step in the DNA cloning process, and this process is referred as ligation. So this ligation is affected by catalytic activity of the enzyme called as DNA ligases. The most commercially used sources are T4 DNA ligase from the phages, as well as the bacterial E. coli DNA ligase. And these enzymes, they make the glue between the broken fragments, or they help in joining of the two fragments by making a phosphodiester bond formation. So these two DNA fragments of a plasmid DNA as well as the insert DNA can be ligated using the DNA ligase enzyme, making a recombinant DNA molecule. So they perfectly serve as a DNA glue, molecular glue to bind the DNA, insert DNA with the cloning vector systems. The third step is ligation of vector and insert, and appropriately we have used two different enzymes. One is the restriction enzyme that is used to cut open the target DNA from the host cell organisms, or the target DNA which codes for a gene of interest coding for an agronomic trait. And also the vector which is used for cloning should also be cut with the specific restriction enzyme, making them amenable for a perfect match of an insert DNA with the vector system. And now let's see how they are being ligated. The vector and insert are ligated together. Once a cleaved vector is going to be mixed with a fragment, insert DNA fragment, which was originally nicked with the same type of restriction enzyme, they can be ligated using the DNA ligase. And we have seen this DNA ligase is a catalytic reaction. So that's again an energy spending reaction which uses ATP molecule for catalytic activity. And they fuse the insert DNA fragment here by making a phosphodiester bond connection. So this ligation of vector and insert can be performed by initially digesting them with the same enzyme. If you use the same enzyme for cutting open the vector as well as the inserts, then we refer that kind of cloning as non-directional cloning. Whereas if you cut with different enzymes, if you cut a vector and insert with different enzyme, we call it as directional cloning, because that gives you no choice option where with a different enzyme, the recognition specificity of the vector and the insert DNA will differ at both the ends, making it a directional cloning. So directional cloning can be done by, it can be achieved with phosphatase treatment, where you can cut the a vector or a fragment, insert DNA fragment with different enzyme, plus you can add a free 5' phosphatases, which will remove the phosphate group at the 5' moiety that will prevent self-ligation of the vector. Normally, most of the commercial vectors which are supplied by the companies, they would be linearized vectors, which are treated with phosphatases. They prevent self-ligation of the vectors until it is used for cloning purposes. Okay, once we have done with the cloning process, we have got ligated our insert DNA into a vector system. We can check for the effectiveness of the ligated product. And this can be visualized by using a biochemical technique called as agarose gel electrophoresis. So here is a summary of this agarose gel electrophoresis, where you can brew the agarose and pour the agarose, molten agarose, along with the dye fragments, or later you can stain them for visualization. The molten agarose is poured in the mold, gel mold systems, and you can insert a comb to create wells for loading of the samples. And after solidification, this casting unit is removed, and the running tray is placed in a buffer system for electrophoresis. So you can load on the different samples in the appropriate wells along with a DNA ladder. Then you can connect it to a power supply unit and do electrophoresis, so that the nucleic acids separate based on their molecular weight, and you can visualize the fragments after staining with any of the DNA visualization stain like ethidium bromide, or you can also go for staining with commercially available cybersafe stain or any cyber green dyes, or even methylene blue can be used for staining purposes. After that, you can visualize and document the gels using a gel documentation system. So here, let's see how the electrophoresed products of a ligation reaction will look like. Here, we have three different lanes. The first lane is the DNA standard markers with a differing molecular weight composition. The second lane is a ligation control sample, where you can see typically two bands. One is of a unligated vector, the other one is a insert DNA. The third lane contains a ligation reaction sample, where by addition of ligase, DNA ligase enzyme, the vector is now fused with the insert DNA. The vector and insert DNA get fused to form a recombinant DNA. So a ligated product will basically has a size larger than 10,000 base pair. In this case, it is larger than 10,000 base pair. That's why they occupy a higher position than the controlled DNA or unligated DNA molecule. The fourth step in DNA cloning or construction of recombinant DNA and transformation process is bacterial transformation and recovery. So what's transformation? It's a process of inducing the cells to take up the recombinant DNA. So the first step here is to prepare a competent cells, which is used to modify the strain of an E. coli, so that that can be made amenable to absorb the recombinant DNA. So these modified strains of E. coli is an apt choice here, where the restriction enzyme system is knocked out in the modified strains, and there is a limitation for recombination system here, and there is a truncation of LPS or lipopolysaccharide, and some of the common strains that are used as a host cell system for bacterial transformation include E. coli DH5 alpha, DH5 DH10 beta, JM101, JM109. These are also commercially available as a host cell system for preparing competent cells or host cells for bacterial transformation. Okay. So do the bacteria naturally take up foreign DNA when it is mixed in a laboratory experiment? Practically no, because not all bacteria take up free floating DNA into the environment, because we know transformation is transfer of a naked DNA without any assistance through a kind of cell to cell contact or aided transformation. It's just take up of a foreign DNA. It's a free floating DNA to be absorbed by the host cell system. So normally the bacterial cells, majority of the bacterial cells are incompetent to absorb the foreign DNA. So we have to make the bacterial cells to uptake the foreign DNA by increasing its competency or make them hungry to take up the foreign DNA. However, we have some bacterial cells which are naturally competent enough to absorb foreign DNA, but that's a limited choice. And those bacterial strains are not normally used in the laboratory for recombinant DNA construction, which includes the Bacillus, Streptococcus, Haemophilus, and Neisseria. These bacteria have natural competence to absorb DNA from the environment. But in the laboratory, in a genetic engineering laboratory, we routinely use a bacteria which is a modified strain of E. coli, basically modified from K12 strain of E. coli, that can be treated with some salts to increase its competency to uptake the foreign DNA. And now these cells are said to be artificially competent or competent cells. So how do we increase the competency? So we can use some of the chloride salts to increase the competency of these cells. The chloride salts can be calcium chloride or rubidium chloride or magnesium chloride. Since the calcium chloride, the calcium ions neutralizes the negative charge on the phosphate backbone of the DNA, because the phosphate backbone has an oxygen moiety that offers the net negative charges to the DNA that would be neutralized by the positive ions in the calcium chloride, so that the DNA is not repulsed from the phospholipid membranes. Phospholipid membrane in the bacterial cells. So this will allow the DNA, our foreign DNA, to get easily passed through the cell membrane into the cytoplasm of the bacteria. So those bacterial cells which are now treated with the calcium salts or rubidium or magnesium chloride salts, they can be made amenable for uptake of foreign DNA, and those treated cells are called as competent cells. So here you can see during the incubation on the ice, the DNA would, the recombinant DNA in our case, would bind on to the surface of the bacteria, and they help in forming a calcium phosphate DNA complex. Now the question is, do the bacterial cells which are treated with the calcium salts by itself make them porous or uptake the foreign DNA directly? No. So that's very limited, and we can increase the uptake of foreign DNA by making some chemical or electric shock process to make the minute pores in the bacterial cells to uptake the foreign DNA. Now let's see the bacterial transformation procedures. Having the competent cells ready in laboratories, normally we prepare competent cells by treating with calcium chloride salts. Once the calcium chloride salt treated cells are ready, they can be immediately used for bacterial transformation. If you want to do at a later date, you can you can store them in a glycerol stock and put them under freezing condition at minus 80 degrees centigrade for about months together. At a later date, you can recover them and you can use it for bacterial transformation. But ensure that if you are going for bacterial transformation, it's always better to use competent cell which is immediately prepared and freshly used, rather than storing them as a glycerol stock and using it at a later stage. Because during storage, the bacterial cell will lose the competency. Now let's see the bacterial transformation procedure. It's a process in which the bacteria uptake the foreign DNA into the host cells. This can be triggered by either applying electric shock or by using heat shock treatments. Now let's see the transformation method. In the first stage, we are going to chemically treat the bacterial cell and give a heat shock to make the foreign DNA enter into the host cell system. Alternatively, you can apply electric shock by using a procedure called as electroporation. The first method here is chemical transformation step, where the cells are grown into a mid-log phase, a very actively dividing mid-log phase, and they are harvested and treated with different salts of calcium chloride. Normally, we use 100 millimolar calcium chloride and subsequently reduce it to 50 millimolar calcium chloride. And every time when we do that, we chill the bacterial cells in ice bath, and the cells treated in this way by using the chloride salts of calcium, magnesium, or rubidium, they are said to be competent. And now these competent cells can be mixed with the DNA on ice and followed by a brief incubation at 42 degrees centigrade. In a water bath, you can appropriately set 42 degrees centigrade for 90 seconds. That would make minute pores in the bacterial cells and allow the foreign DNA to enter into that. Here, this is a quick flow of the ligation reaction and a controlled reaction. Those cells are used for transformation, ligation and transformation. So here, once you prepare a competent cell, you can go for mobilizing the ligated DNA as well as to compare a controlled DNA can be mixed with the host cells. So they can be incubated in ice for 10 minutes as a prior to transformation. The bacterial cells and the ligation mixer has to be chilled in the ice for about 10 minutes, and then you can incubate in a water bath at 42 degrees centigrade for 96 seconds. And soon after that, you bring the cells, the transformed cells, into the ice bath again for 2 minutes. And now it is assumed that the foreign DNA has been mobilized into the bacterial cells, and these cells which are harboring your recombinant DNA is now has to be allowed for multiplication or propagation in by incubating them in a water bath, or you can use a shaker system for allowing them to multiply. So this is normally done by adding a rich medium of the LB medium or SOC medium is added, which will allow a quicker multiplication of the foreign DNA or the recombinant DNA into the host cells, and subsequently they can be put for screening in an appropriate antibiotic selection system. The second way to transform is by applying electric shock. So this method is called as electroporation, where the cells are normally grown to a mid-log phase, and then they are washed extensively using water to eliminate these salts. So these cells, which are washed with water to remove all the salts, are going to be mixed with the ligated DNA, and they're going to be transformed by using an cuvette. They have a cuvette-like system which would be connected to electrodes, and upon application of a short electric pulse at about 8 kilovolt per centimeter, the cells which are applied with the electric shock would cause small holes due to the short electric impulse in the cell membrane that would allow the foreign DNA or a recombinant DNA to enter into the bacterial cells. Here you can see the comparison of chemical transformation and the electroporation procedures. In the case of chemical transformation, we have seen that the bacterial cells are made electrically competent by treating with the chloride salts of calcium, magnesium, rubidium, and then they are going to be mixed with the recombinant DNA, ligated molecules, and apply heat shock at 42 degrees centigrade for 90 seconds. That would allow the recombinant DNA to enter into the porous bacterial cell wall, and they can be subsequently recovered. In the second step of bacterial transformation, applying electric shock, the electrocompetent cells are prepared and they are incubated in an appropriate environment. When you apply a short impulse of electric shock, that would make a minute pores in the bacterial cells and allow the foreign DNA to enter, and subsequently they can recovered in a rich medium by incubating at 37 degrees centigrade. So this animated image would show you how the ampicillin sensitive E. coli cells get mobilized into the bacterial cells. So the recombinant DNA enters into the host cell, they remain in the cytoplasm, and subsequently they can be used. These transformed E. coli cells can be deployed for screening purposes to choose whether they possess the recombinant DNA or not. The next step is recovery of the cells after transformation event. So this step will allow the cell to regain the strength and start to multiply. And this step is necessary for the cell to allow the expression of the antibiotic resistant gene present on the plasmid, because we have seen it's an ideal feature of a plasmid to possess a selectable marker and antibiotic resistant gene which aids in the screening process. So for the gene to produce enzyme to degrade the antibiotic, we have to allow sufficient time. That will also be the time required for the cells to multiply the cloned DNA fragment in a larger number, propagate them in a larger number for subsequent steps. So this is what we call it as recovery period. So we would be adding about 250 microliter of Luria betani broth to both the control tubes as well as ligated tubes using a sterile pipettes, and they can be incubated in ice for initially, and then moved on to 37 degrees centigrade water bath for about 5 minutes. And subsequently, in a recovery step, after incubation, they would be plated onto a medium containing antibiotic. Say, for example, if your recombinant plasmids contain ampicillin resistant gene, which produces an enzyme called as beta lactamase, that would degrade the beta lactam ring of ampicillin and allow the recombinant cells to survive. So when you plate the bacterial cells onto the media containing the Luria batani media as well as the selecting agent ampicillin, only those cells which carry the resistant genes of an antibiotic will allow the bacterial cells to survive by degradation of the ampicillin by the beta lactamase activity. So here, this is a comparison of the control experiment and control tubes and the experimental tubes. In this case, you are going to ligate and transform a bacterial cells without an insert, and then they are plated onto a medium without any antibiotic. There is no ampicillin in the growth medium. This will allow the cells to grow because there is no selection pressure deployed in this case. But if the control cells, which do not possess a recombinant DNA with an ampicillin resistant gene, when they are plated on a media containing ampicillin, that would kill the cells and they will not allow it to grow. So this is how a control experiment is visualized. Whereas in case of experimental tubes, where your recombinant DNA, which contains a resistant gene of an antibiotic in a plasmid, also has your insert DNA. When it is plated on a medium containing medium without antibiotic, naturally it will allow the cells to grow because there is no selection pressure applied here. Whereas when there is a recombinant plasmids added to that, which contains the ampicillin resistant gene, which would degrade the antibiotic enzyme in that, this would allow the cells to survive by degrading the ampicillin antibiotic. So the ampicillin resistant gene produces a beta lactamase and allow only those cells recombinant DNA which process the resistant gene to survive, whereas those cells which has not a recombinant DNA will die in the selection process. The fifth step and the last step in the cloning process is selecting and differentiating the cells as a transformant or a non-transformant, or the DNA, a recombinant DNA or a non-recombinant DNA. This is purely based on visualization experiments. So here we use blue-white selection process or blue-white screening of recombinants. So to deploy this blue-white screening process, we need to clone the DNA fragment in an MCS region. So here you can see we are choosing a vector system which contains a LAC-Z gene. The LAC-Z or LAC-Z gene codes for the enzyme beta galactosidase. You can recollect your lac operon concept, where there are three structural genes, lac-Z, lac-Y, and lac-A. Among these three, lac-Z codes for beta galactosidase, which would break the lactose and convert into simple sugars, glucose and galactose. So the same lac-Z gene is cloned in the DNA fragment here, and this lac-Z gene has multiple cloning sites, which is used for cloning the foreign DNA fragment. Once you are inserting a DNA in the lac-Z region, you are practically you are inactivating the lac-Z gene due to insertion, and this phenomenon is what we refer as insertional inactivation. Because of insertion of foreign DNA in the lac-Z gene position, you are inactivating the functionality of the lac-Z gene to code for the beta galactosidase enzyme. Whereas here you can comparatively can see another vector which do not carry your DNA of interest, and this is just a self-ligated vector which contains the lac Z, and this is a functional complement of that. Once these plasmids are mobilized into a bacterial host system, they would show a differing level of expression of genes, and accordingly we can visualize them in a blue-white screening process. So for this to take place in a laboratory experiment, we use a colorless substrate called as XGAL, and this XGAL would mimic the lactose analog in a lac operon concept. So this XGAL is a X-galactoside substrate molecule, which upon action by the beta-galactosidase enzyme produced by the LAC-Z gene will convert the colorless substrate into a colored product called as 4-chloro, 3-bromo, indigo, blue-colored product. So those cells which expresses the LAC-Z gene complement will produce the enzyme and allow the degradation of the colorless substrate into a colored product, so that you can see the blue color colonies here. But if the foreign DNA is inserted into the LAC-Z region, I told you that makes inactivation of the insertion, the LAC-Z gene due to insertion. The phenomenon is referred as insertional inactivation. That makes the LAC-Z inactive, so the beta-galactosidase is not produced, meaning that because of absence of beta-lactamase activity, the colorless substrate remain as such without conversion into a blue-colored product. So those colonies which are blue in color are the one which has functional LAC-Z activity, and they do not possess a insert DNA. Whereas the white color clones are one which uh, the blue color one is a colony which has active beta galactosidase, and it produces a blue color colony because of production of the beta galactosidase enzyme. Whereas the white color colony is supposed to contain insert DNA, because presence of insert has inactivated the beta lactamase activity, and there is no conversion of the XGAL into a colored product. That's why the colony remains white in color. So that's how we can differentiate. And here you can typically see the blue-white screening of recombinants. So whatever the cells that are observed in white color, they are said to be positive, and they are supposed to contain your insert DNA in the recombinants. Whereas those cells which are blue in color are the one, they have a functional beta-lactamase activity, and they do not possess your insert DNA in the recombinant molecules. So that can be visually observed in a screening system like this. So this technique is also called as alpha complementation assay or blue-white screening system for selection of recombinants. We can also go for checking out the presence of foreign DNA in your recombinant DNA by isolating the plasmid DNA from the recombinants. So to do so, we have to culture the bacterial cells in an appropriate broth system. Here you can, normally we use a LB broth or a Luria betani broth, allow the cells pick up the white colonies from the blue-white plates and grow them overnight. And you can subsequently isolate plasmid DNA from them by adopting alkali lysis mini prep method. This alkali lysis method is very popular, and it was reported by Birnbaum and Dolly in 1979. And this uses three different solution systems: solution 1, solution 2, and solution 3. The first solution is a suspension buffer, also called as TEG buffer, which contains tris, EDTA, and glucose. The tris is basically a buffer which stabilizes the pH, whereas the EDTA being a catalyst ion which chelates the metal ions, and it weakens the cell wall. And the glucose serves as an osmoticum that maintains the osmotic pressure so that the cell is intact during the entire process of isolation of bacterial plasmid DNA. Once you grow the cells overnight and harvest them, you are resuspending the cells in the solution 1 or a TEG buffer, making the cells now in a condition amenable for lysis. In the subsequent step, these cells are going to be treated with a solution 2 that primarily contains a detergent, an anionic detergent called a sodium dodecyl sulfate, along with an alkali, NaOH. In an alkalized environment, the SDS is going to be breaking the cell wall of the bacteria. Here you can see the bacterial cell wall is broken, and they release the cellular component as a cell extract. In the subsequent stage, So during the course of the treatment with solution one and solution two, the solution one, the tech buffer is of pH around 8. And when you treat it with alkali lysis, the pH would rise up to around 12 to 14. And in the subsequent step, you have to neutralize or bring down the pH to what it was earlier so that the DNA can be intact. So you are going to bring down the pH in the subsequent step by neutralizing them with an acetate molecule. So that's what is called a solution 3 or potassium acetate, which would bring down the pH and allow the intactness of the plasmid DNA to be isolated in the subsequent process. So finally, you would get a pure nucleic acid of plasmids, which can be electrophoresed and check for its quality. And these isolated plasmids can now be subjected to restriction endonuclease treatment that would break the plasmid to release the insert DNA. So the presence of insert would confirm that it's a recombinant DNA. You can make a comparison with the uncut vector, as well as you can make a cut and check with a DNA standard or a ladder to ensure that your insert is present in the recombinant DNA process. Alternatively, you can also check for the presence of insert by doing a colony PCR. So once you grow your cells, bacterial cells harboring your recombinant DNA overnight, you can just use a toothpick to pick up a colony and just dissolve in 20 microliters of sterile water. You dissolve the bacterial cells. You can directly use this as a template DNA and amplify in a PCR reaction in a thermal cycler with gene-specific primers. So the amplification will ensure you that the host cell is harboring the recombinant DNA. Normally, we used to get confused or often mistaken with these terminologies, that what is a transformant and what's a recombinant, or how do we classify a transformant from a non-transformant, a recombinant from a non-recombinant. So if you say transformant, the cell that have taken up foreign DNA are called as transformant. So transformant is with respect to the host cell. So those host cell which has taken up the foreign DNA is said to be transformant, and the cell that do not take up the foreign DNA are referred to as non-transformants. Whereas when we compare recombinant with a non-recombinant, this is with respect to the plasmids. So the cells that has a foreign DNA inserted into their genome are called recombinants. Means the recombination event has taken place, meaning that the insert DNA is mobilized. Insert DNA is cloned into a vector, that's a recombinant, and has been mobilized into a cell during transformation process. So the presence of foreign DNA integrated into a genome will confirm them as a recombinant present in the host cells. Whereas a parental genome which do not contain the insert DNA are referred to be non-recombinants, meaning that only a self-ligated vector without a insert DNA has been mobilized into the host cell, and they are said to be a non-recombinant. So for easy understanding, transformation is with respect to the cells, recombinants is with respect to the vector systems that present here, in this case plasmids. If they do not possess the insert, then we say them that they are non-recombinants. Okay, once we are good at transformation, and we can also work out how efficiently our transformation process was carried out. Remember, the transformation is basically a technique where it's having a poor success, or it's an inefficient process, in the sense only 1 in 10,000 cells will take up the foreign DNA or possess the insert DNA upon transformation. So we have more number of cells in a colony of mixture. When you do away with the transformation experiments, that could somehow correlate or improve the efficiency of transformation. But however, invariably the transformation is a highly inefficient process. So how do we then check up how much is the transformation efficiency? This can be done by counting the number of colonies on each plate after incubating them in a recovery process. You can incubate the bacterial cells grown on a medium, and you can check for the number of colonies using a colony counter, or you can make a quadrats of the bacterial petri dishes and manually count the cells, and this would help you work out the transformation efficiency. So what is the transformation efficiency? So transformation efficiency would represent the number of colonies observed, the transformed colonies observed per microgram of the plasmid that's been used for construction of recombinant DNA and subsequently mobilized into the host cell. So this has got a formula, that's number of transformants per microgram is equal to the final volume at recovery divided by the volume that's plated onto a medium into the number of transformants observed divided by the microgram of DNA, the insert DNA. So that would tell you how much is the transformation efficiency. As I told you, the transformation efficiency is a highly inefficient process. It generally takes about 1 into 10 power 4 to 10 power 8 cells per microgram of plasmid. That's what is the realized transformation efficiency in any laboratory experiments. So we have seen in detail about the reconstruction of recombinant DNA and bacterial transformation. You would also get to see what are the different applications this particular recombinant DNA technology has. So it's in summary where you can see from a host DNA, you can isolate the plasmid from the bacterial cell, and these bacterial cells can be treated with an restriction enzyme to cut open, and from a target organism, you can isolate the genes of interest, which may be coding for a particular trait of interest. These genes can be again cut with the same restriction enzyme, and they can be mixed with a cut vector system and ligated, so that will create a recombinant plasmid DNA. So here you are using two enzyme system. One is the restriction enzyme for cutting the DNA and ligase system for joining the DNA to make a recombinant plasmid or recombinant DNA. So this recombinant DNA is going to be mobilized into the host cell for propagation, and this is what we call it as bacterial transformation. Once the cells are cloned with the gene of interest into the host cell system, in the subsequent step, you would identify the desired clone which possess your recombinants or the transformants which is expressing your foreign DNA. And after the screening of your recombinants, these cells can be upscaled to produce the industrial products or an antibiotics or whatever recombinant proteins you have for which the gene has been inserted into can be exploited on a larger scale in a commercial procedures. So what are the applications of this transformation? This transformation can be even can be used to clone the DNA to create transgenic plants that express genes for the pest and disease resistance. So here there's an example of creating transgenic plants. A good example is your BT cotton that harbors the genes of cry protein that would impart resistance to the Lepidopteran pest. And here, the copies of the genes which produce the recombinant protein can also be deployed for conducting basic research to understand how the protein synthesis is involved, and the enzymatic activity can be studied from the recombinant proteins that are produced. So the recombinant proteins which are specific for cleaning up the environment by for removal of the toxic waste can be done. So you can also deploy the recombinant DNA technology or a transformation process to clone genes which can produce protein products that would degrade the toxic waste in the environment. In the medical sciences, the proteins which would dissolve the blood clot, like thrombins, can be produced, which aids in the therapeutic procedures in the medical sciences. And some of the industrial proteins which aids in the human growth hormone production can also be produced using a bacterial cell as an expression system. So invariably, this recombinant DNA technology is a potential tool which can practically amplify any cloned DNA in its host cell system that would produce a recombinant protein of interest with multivarious applications. And this can be in agricultural sciences. We deploy primarily for producing the transgenic plants with improved agronomic traits. That's it. Thank you.